The Conversational Flow, a KCAS podcast.
Hello, and welcome to the 14th episode of The Conversational Flow, an official podcast of KCAS Bioanalytical and Biomarker Services. KCAS is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. Unlike KCAS' other podcasts, the Weekly Bioanalysis, which features news and topics more generally associated with the world of bioanalysis, the Conversational Flow focuses more on the niche of flow cytometry and its role within the greater industry. My name is Adam, and I've been in the flow cytometry space for over 15 years, and I'm here with my co-host Brian.
Thanks, Adam. My name is Brian. I've been in the flow cytometry space for over 13 years.
Brian and I are members of the growing leadership and scientific advisory team at KCAS. Either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS services. While our KCAS headquarters are located in Kansas, with additional locations in Europe and a partner location in Australia, we're both here in Philadelphia, and Jeremy, our producer, is in Missouri. So we're truly all over the map. We're exhilarated to have you listen to the 14th episode of our podcast, which will be available virtually everywhere. Wherever you choose to find and play your podcast, you will likely find the Conversational Flow. We welcome all of you, whether you're joining us for the first time or if you're a regular listener to any of our other podcasts. Today's episode will be a review of the latest news and resources and a focus on a topic of our choosing before discussing any feedback we've received from you. We're constantly looking for topics, and we'd be happy to discuss something that you want us to cover. So again, we're ecstatic to have you here, and we look forward to having a fun episode today. To kick us off for episode 14, Brian is going to go over some news and resources before diving into today's topic. Brian?
Thanks, Adam. We do have a packed agenda today. We've got some medical investment and KCAS news and an exciting paper about backbone modifications.
You know, it just occurred to me, though, you say we have a packed agenda. I think you say that every time. So we really do have a packed agenda, but just wanted to point that out. We have so many different words that we use for the intro, so maybe we need to go, full or I don't know. Sorry, a little off track.
For the first item in our medical research news, I've got here the FDA approval of a new therapy for adults with relapsed B-cell ALL, so acute lymphoblastic leukemia. I have specifically avoided saying the name of the approval as it is quite long and I don't want to butcher it. But it's a new therapy from Autolus. The trade name will be Aucatzyl, which I'm not sure is much better, but it's...
Okay, I'm going to go and try this. It looks like it's obectogene autoleucel. All right,
Okay.
Close, but I still know I messed that up a little bit. Now, I will say, in reading this, definitely very interesting here about the approval. We've talked about these types of therapies before. And as I'm reading through, I saw that the clinical trial name is Felix, right? And we've worked with plenty of different clinical trials, and they've all got different names. And, you know, some of these are really inspirational names, and others are descriptive, and others don't generally seem to have anything to do with the project itself. And so when I saw Felix, I'm like trying to understand, are there rules around clinical trial naming? So what's your experience there, Brian?
My understanding is that, yes, there are. And if I remember correctly, there is a very small organization that is responsible for coming out with these names. So I'm going to have to look that up and bring that up on the next podcast.
And evidently, though, it happens very early on. So Fierce Pharma had an article specifically what's in a name inside the process of naming clinical trials, right? And so that's where I'm pulling my information. It can be an acronym. It can be inspirational. And it doesn't even necessarily have to be associated with the therapeutic. And so it's kind of loose as to what it is, right? But the interesting point that I found out about was that there is even some discussion, some groups would prefer not to have these clinical trial names be super upbeat, super positive, because it almost draws people into it. Now, the counterpoint to that, I will say, is that you're drawing in people who need this therapy, right? So it's not like you're taking somebody off the street who doesn't need to be in a clinical trial, and then all of a sudden they see the name and it's inspirational, and like, I got to be on this clinical trial. So, I get both sides of it, but it was just really interesting. And I know a little bit sidetracked from the specific news, but really interesting to see kind of that part of clinical trials.
Agreed. And I think it is an interesting point, right? It's basically bringing marketing into areas where marketing isn't traditionally seen or allowed, and apparently there's some questions about it, right?
Mm-hmm.
And you could say the same thing about drug advertisements in general. Like, do we need to have TV ads with drugs on it? Towards your point, ideally, the people that want this would go and talk to their doctors and decide with them. So, maybe there's some benefit to it with people that might not otherwise know it's available. So yeah, it is an interesting point.
Well, and interesting you say they might not know. Before getting into this industry, I don't really remember, and maybe it was just because I've been in the industry so long that previously there weren't as many commercials for pharmaceuticals online, or the information out there for clinical trials. But I just don't remember seeing the names so much, and just more and more. Obviously, now each clinical trial needs to have a name. But it's interesting to the counter to you saying that it leaves it in your mind. I just never had that before.
Yeah, no, you're absolutely right. I don't think that'll be the case with the full name, right? The obecabitinin autoleucel.
Nice, you got it.
I mean, by the end there, right? Yeah, indeed. So that's probably not going to stick in people's mind, but definitely the trial name. Mm-hmm. And I guess the important part about this FDA approval is, this is another CD19 genetically modified autologous T cell therapy. So basically, they're taking cells from a patient, doing basically CD19 gene modification, and then putting them back into the same patient.
Right. And as you mentioned, ALL, so acute lymphoblastic leukemia, a disease that progresses very rapidly in the blood and bone marrow. And essentially what happens is these cancer cells crowd out healthy cells. And so it is a very important area of focus. And, I know we've talked a lot about CD19 specifically, and that targets B cells, but it is even higher, more highly expressed on these tumor cells, and so that's what makes it an attractive target. So anyway, proceed.
That's it. Yeah, I mean, with this FDA approval, they did talk about some of the side effects that were witnessed on the study. Interesting to me, and something I'm continuing to track, is the amount of basically chemotherapy or immune suppression required either prior or after the administration of these T cell therapies, especially as we talk about the migration of cell therapies into autoimmune diseases. These types of interventions are sort of par for the course and expected with oncology, but definitely not with less severe autoimmune disorders. So I'll be interested to see how that is modified with time, and as we sort of get into newer generations of cell therapies, is that still going to be required? In this case, it definitely was, that they needed to knock down a large portion of the immune system prior to administering this, and we're very concerned about the cytokine release syndrome still.
Well, and it brings up how can you use an anti-CD19 for cancer, but then you can also potentially use that in an autoimmune condition, right? And so what we mentioned was a lot of times when you're doing cancer therapy, you're looking for some sort of protein that's only expressed on the tumor cells and not off target. And the reason being is you don't want to have some sort of side effect because you're hitting some other cell type or tissue and whatnot. Here, the CD19, it does have that off target because CD19 is part of healthy B cells as well as the tumor cells. But what can happen is it can destroy all those cells, and then you can have the B cells. The B cells do come back, right? And so you can have a healthy immune system still, but you still have a functioning immune system, not in total, after losing the B cells, the CD19 positive B cells. what happens with autoimmunity is the B cells generate antibodies that then attack healthy tissue, right? And so by targeting those B cells, destroying those B cells, the thought is, and this is very general, right? So there are plenty of different paradigms of cell therapy and different targets, but here, looking at targeting CD19 B cells to then remove them and essentially reset that attack, reset the immune system so it's not attacking the healthy tissue anymore. And there have been successes with the therapy in that area.
Yeah, absolutely. As you say, a lot of parallels, or at least, you're within the same mechanism of action, and cancer and autoimmune disorders are sort of just different areas within the spectrum of that mechanism, if you will. So, always interesting to see how these cell therapies are getting approved. Exciting, and congratulations to the Autolus team for this approval, and looking forward to a number of others in the near future. Switching over to investment news, the first two stories I had come across were a little bit more negative, so I included 3 this time instead of 2. The first one was AbbVie's Cerevel buyout. And the reason, and this is basically the fact that Cerevel is testing out a new schizophrenia drug, and it was in 2 pivotal phase 2 trials. And just as a reminder for the audience, the phase 2 trials are where the drug demonstrates its efficacy, so its ability to actually impact the disease indication that they are trying to impact. And this drug failed those 2 schizophrenia clinical trial tests, which is pretty significant because in a lot of buyouts like this, pharma companies will try to buy a de-risked asset. So that means they were really expecting this drug to, be safe for people to use. So it passed its phase 1. So, checkbox there. But then also that they had pretty high confidence that it would pass its efficacy test and then go on to be sort of a new blockbuster type drug. This one was slated to compete directly against the BMS acquisition that was recently done as well.
Right, that's Cobenfy for BMS, right?
Correct.
And so this acquisition was just finished in August of this year, so just a few months ago, and that was $8.7 billion, I think you had mentioned. Interestingly, with this, one of the reasons put forward as to why this may have happened was they had a higher placebo effect than expected that may have caused the miss. And this is always something that's a very interesting discussion, is the placebo effect. And there had been some data, I'll be honest, I haven't looked into it very recently, but I remember a couple of years ago that there was a trending of higher placebo effect. And so for those that don't know, essentially a placebo effect, you take a sugar pill. So part of the study gets the treatment, part of the study gets essentially a sugar pill. And you have to track both parts of the population to make sure that you understand what is happening, that the treatment is working as intended, that the positive impacts are from that treatment and not from something else. And so here, specifically, that placebo effect was higher. Now, certain things that could relate or could drive a higher placebo effect is the use of patient-reported outcomes. So as you get the information from patients, if that's the case, maybe the patients just were in a different part of their life and, things seem more positive. There could be a million different things that might drive it some. And so studies don't just rely on patient-reported outcomes. But then the number of trial participants, obviously, or the trial length could also play a role. If you have too few participants, that by in and of itself just could drive that percentage up. Or it could be issues either with sites or endpoints of the study. So I found that really interesting just because I've always watched or tried to read a little bit about the placebo effect and why it might move over time.
It is very interesting and very near and dear to our hearts with flow cytometry, right? Because we, of course, do quite a bit of testing on in some cases, healthy donors, and just the variability there and changing dynamics within the population, have an impact on a lot of our testing as well. But you're absolutely right that there was this higher placebo effect and the results were relatively close. It gives AbbVie the chance to go back and redo another phase 2 trial. But even if they redo the phase 2 trial, then it's just more money, of course, to run the trial, but also time. And time is always of the essence for these large pharma groups, especially as they're looking at patent cliffs and other competitors on the market. So definitely a surprising result based on the buyout and the amount of investment there. But as you say, it will be interesting to see how that proceeds forward, and hopefully will give subjects suffering from schizophrenia other options in the near future.
Now, I was curious, how much do you know in the area of antipsychotics and the mechanism of action specifically here? So what they're targeting. I'll be honest, I wanted to dive into a little bit more of it, and I do have it tabbed for future reading. But noting that, for example, Cobenfy is an M1, M4 preferring muscarinic agonist, while emraclidine, which is the one that had failed here, is an M4 muscarinic receptor agonist. Are you familiar with anything there?
I am not very familiar with much of neurology, right? Like we try to stick to our immunology niche here in flow cytometry as much as possible. So for these specific receptors, no, not as much. Mm-hmm. No, there's a couple competing theories about exactly what induces schizophrenia, and very familiar with muscarinic receptors in general, but not as familiar with the specific chemistry of these two drugs.
Right. It's interesting just because of the difference. Obviously, one's an M1, M4 versus an M4. And so when you see data like this, the movement, and so here we're back into investments, companies that are focusing on an M1, M4 versus an M4 specifically, or other versions. Obviously, news like this drives the market. And this was no different, where BMS's shares shot up 13% last week, so the week of November 11th. And then AbbVie's was down 12% following this news.
It is surprising. I understand that these are blockbuster drugs, but it's surprising that so much of the company's share price would be driven by these types of tests.
Especially when you're not talking about small companies by any stretch of the imagination, right?
Right? Yeah. I mean, if we're talking about like a 1 or 2 molecule company, then yeah, sure, of course that makes sense. But for these giant pharma companies that are diversified into multiple indications, I was a little bit surprised by that.
Same. Yeah. I never would have thought you'd see 13% change on a BMS and for something like this. Yeah.
Yeah. Well, that does lead us potentially into our next discussion of 23andMe. So 23andMe, I wanted to cover just as an example of a service provider in the industry. And here, 23andMe is fallen on rough times, and has definitely lost a lot of their stock value, but is now also needing to cut portions of its workforce and discontinuing its therapeutics division.
So you mentioned falling on tough times. Looking in the numbers, and I will say, hopefully I've got these right, did do a little bit of digging to try to get this correct, but they went from a valuation in 2021 of $6 billion. As of September, that was at $150 million. And so that's hard times. A little bit of a drop here or there. That's quite a bit more. And I mean, to that point, they ended up cutting about 40% of their workforce, which equates to 200 employees or so. And you hate to see stuff like that. Now, what they had chosen to do was to try to develop their own research programs, their own drugs, clinical trials. And that's what they're now moving away from. And, the thought behind it was, this is a genetics company. They offer, that sequencing. So you get an idea of heritage and diseases, things of that nature. But what they struggled with is that people would send it in and they only have to send it in once. It's not like you constantly had to send in for new sequencing or something like that. And to have have to take that next step, they wanted to look into doing their own research, doing their own drug development. But at least to this point, it hadn't produced enough benefit to lift the company away from this current point.
That's it. And, the reason I wanted to cover it was because people talk about flow cytometry, especially now that we can do high dimensional flow cytometry, sometimes in the same vein, where a lot of people want to have, a single panel that they can use all over the place, and they will always give you, all the answers. Maybe you look at it for one answer initially, but you can go back to it for all of the others. And to some extent, that's sort of 23andMe's model as well, where they ideally would have run a single sequencing assay and then been able to use the data in umpteen different ways to give them different sort of answers to the same question, if you will. Or be able to query the same answers multiple times for multiple different research purposes.
Well, it is amazing, though, the difference in the amount of data. So I, a couple of years ago, had experience going through and having to do genetic analysis. And a lot of the places will offer panels and several options, and you pay for those options. I guess it's kind of a pool of things that they're testing in certain areas. But what later found out was then we wanted to look into other things. They've already got the other data, at least, and this may not be for every company, right? But at least the experience that I had was basically pay for more, and then they were able to give us more data. And so 23andMe, Ancestry.com, things like that, to your point, it's that there's so much information that's provided, but it's a one-off thing, and it's very nice. And I think with some of the ways that they operate, Ancestry.com gives you information on how connected you are, so on and so forth. We've all seen the articles of people who have reconnected with family members that they didn't even know. And it gives me the thought of somebody switched at birth, right? You find out that your parents aren't your parents, at least biologically, I'm saying. So I'm going completely far afield, but...
No worries. I guess my driving point was, yes, in some cases that works out, right? Where you can do one test and get a number of answers and come back and, as you say, maybe you pay for data analysis on one answer one time and then get data analysis on another answer another time. Mm-hmm. But I think what eventually got them as well is that they hadn't validated these tests for some of the different endpoints that they were using either. Mm-hmm. And while, yes, some of this information is fine just as a surface level run across multiple parameters there, some other pieces really needed to be validated. And it's not unlike flow cytometry where, again, the 40 color panel will be great in a healthy donor. Do you need to maybe requalify that for disease specific matrices? Or do you need to look at it in, some sort of ALL model? Do you need to look at it again in some type of autoimmune model? In some cases, I think that answer is yes.
Now, correct me if I'm wrong. Are you queuing up the paper? Like, we still have one more investment thing. So
there's more. Don't worry.
There'll be more coming before we get to the paper. But you're totally talking about what we'll go over in the paper.
That's it. And that's why I thought this news article was perfect because, again, about this. How global can any omics approach really be versus how local does it need to be? So something to be covered in just a few moments too.
Yeah.
All right. And then the last investment news I had was on Quell Therapeutics. And they recently achieved a milestone for a CAR Treg cell therapy program for type 1 diabetes.
Yeah, very interesting use of CAR here. You've got your type 1 diabetes, usually more of that early onset, so the autoimmune condition where you have cells attacking the insulin-producing beta cells in the pancreas versus type 2, which is more characterized towards being overweight, having high blood sugar, and so having more of an insulin resistance. And so this specifically would be for type 1 diabetes.
That's correct. And, CAR Tregs are, of course, near and dear to our heart because we work with so many cell therapies and some of them are indeed CAR Tregs. So was excited to see about this sort of new therapy type being applied to diabetes. Interesting to think about sort of a single shot treatment that might be able to address a decent portion of this disease.
Right. And it's different than a lot of the other news articles that we've seen as far as the treatment of type 2 diabetes, the GLP based drugs, things of that nature.
That's true. This is so. Of course, type 1 diabetes is much more of sort of an autoimmune disease that has the early onset and things like this. And a lot of the therapies for type 1 diabetes are how can we implant new beta islets later on, and can we get these beta islets to produce insulin, and can we get them to survive in the system? And in many cases, that answer has been no.
Right.
So having this type of therapy either on its own or potentially in combination with one of those others may lead to a better chance of success, where, what's driving the failure of some of these other implantation procedures is the initial cells that were killing the beta cells in the first place might still be there. So if you can put in these CAR Tregs, can you reduce those?
Right. And so this is AstraZeneca, right? And really, again, kind of an unmet need. And so we'll keep watching closely to see how it progresses.
That's it. Yeah. Exciting times. Also in exciting times, in terms of KCAS news, we do want to welcome Carla Mancata, who will be the new Flow Cytometry business director for KCAS Bio. So excited to have her on board.
Oh, very much so. I got the chance to meet with her just last week and very impressed. I know that she's got a lot of experience in the industry, a lot of connections there, really good thoughts around flow cytometry, and it comes highly recommended from many friends outside of KCAS Bio. So really looking forward to having her on the team and seeing what she can do.
Yeah, absolutely. And with that, probably a good segue into the paper of the month.
Yeah, so we're looking at flow cytometry assay modifications, recommendations for method validation based on CLSI H62 guidelines. And so this is, oh man, did I not write that down? I know we always go through the impact factor, and it has an impact factor of 2.3. And with that too, let me make sure that I've got this pulled up. We are in clinical cytometry. So it's clinical cytometry. The impact factor is 2.3 there. And it's a very important publication within the realm of flow cytometry, as you might imagine. The paper was written by many within the field, so a very cross-collaborative paper here. And so really aiming to continue to provide guidance around validations, which is something that, as the paper mentions and the title mentions, so H62 was released in 2021. CLSI, what is that? That's the Clinical and Laboratory Standards Institute. So it's not for profit and develops lab standards worldwide, right? And so made up of flow cytometrists from AAPS, which is the American Association of Pharmaceutical Scientists, and ICCS, the International Clinical Cytometry Society. And for this, this is here H62, not the paper itself. H62 was put together by a 37-member committee where they had at least two of the professional arm of the industry, professional industry and government. So really trying to cover the breadth of experience here. But one of the things I will say is they do caveat it right in the beginning that these are recommendations, but because of the wide array of tests that are out there, it's really hard to specifically say this is absolutely how you have to do it. And so what you have to do is read through and see where do things fit. How does this make sense for the assay we're trying to do?
Absolutely. And this paper has a little bit of everything, right? It's got regulatory guidelines, it's got flow cytometry, it's got changing things, and also some real world implications. So definitely a good one to explore for the podcast.
Yeah, and the thing that I really liked was seeing case studies in there, because it's one thing to get the recommendations. And, a lot of times, what kind of recommendations are you looking for? Well, if I do this, what do I need? What is enough to be able to stand on to say that my assay is validated? And that's going to be number of donors. What kind of test scripts do you want to run? Right? It's really important to define these things so that later on, when somebody comes in and says, "Well, you did this, but you didn't do this," you have a leg to stand on. You say, well, that really doesn't make sense for this assay, right? And they did break it down. So if you're transitioning to a new cytometer, if you're changing your antibody, and multiple things that you could do there, right? You can change the clone of the antibody, but keep the same fluorophore. You could essentially keep the same antibody, but get it from a different vendor. So it's the same clone and same fluorophore. How do you then go about validating that? And two, what is the use of your assay? So is this going to be something where your secondary exploratory endpoints is a lot of the studies that we support there? Or are you going and doing MRD, for example? And so you might need to have a bit more stringency. And what I mean by that is you might add more donors, you might add more test scripts to it.
They did definitely do a good job of exploring that and then categorizing it into risk levels and categorizing some of the investigations you might want to consider. In terms of how to, I guess, go through the paper, one of the things that we had touched upon was, can we review this paper in the context of a backbone panel?
Mm-hmm.
And what is a backbone panel? It's one that we basically make with the express intent to change it later on. Right? So it's basically the starting point for all other panels that you'll develop later on.
Right. And so basically, what you're looking at is you've got maybe 75%, 90% of the panel designed enough that the client is coming in and they understand what it is that they're getting, but they may have other interests, right? So you've got a panel that interrogates T cells and B cells, but there's some sort of niche marker that the client is interested in. Now, when we do a backbone panel, that niche marker isn't going to apply to everybody. And so not everybody wants to pay for that marker on that panel. And so a backbone panel isn't going to have those specialties in it. It's going to have that core that gets you started, right? And then the client comes in, we leave certain channels open. And why do we do that? Well, those generally are the channels that may be the most important ones to the client, right? We know that we can get really good resolution off of all of these other markers with the other channels that we're using, the other fluorophores. We leave a couple of really strong ones, maybe the PE, whatnot, and allow the client to then put their marker on that so that the panel then really works well in interrogating everything and looking at their target of choice.
Right. And, I think what this paper does in the context of backbone panels is it gives you a very good framework on what to do. The first thing, ideally, we wouldn't need to do this very often, but they talk about how do you move a panel from one instrument to another. But then more importantly for backbone panels, at least, is once you have this backbone panel, what is the recommendation after you modify it? So you mentioned a couple things, Adam. You mentioned, can we add markers in? You mentioned, are the fluorophores optimal? So if we need to change fluorophores on the panel, what does that look like? You mentioned, I think, what if we need to actually take a marker out of the panel? Mm-hmm. Or what if we needed to change the clone? So what if we had one clone of CD19 and we need to change to another one based on epitope availability on the CD19 marker?
Right. And one of the key things, I think one of the simplest aspects they mention on were reportables not changing. So as long as your reportable set is the same, then it's kind of an easier decision, less effort, I would say, has to go into it than if you all of a sudden you're introducing markers that are introducing reportables. And so that with a backbone, though, one of the things that would do, it's great if you want to have something that's optimized, right? You basically, what we would do, you have the backbone panel. It's going to save you on titrations because we've already done all that work for you, with the exception of the one that would get added, right? The marker or markers that would finalize the panel. So you save money there. The optimization, it's fairly straightforward. You just run and make sure that you're getting the data, and you could then interrogate your samples whatever you want. Now, if you want to go the next step, you've got qualification, right? And that's what you're talking about, qualification or validation, where you can then make sure that the assay is performing to a certain standard. One of the other parts of this too is whether or not you're looking at qualitative reportables versus quasi-semi-quantitative reportables. And so those are things that are part of the consideration. And so if you are using an output such as frequencies, right, that's one thing. But if you're looking at MESF, for example, then all of a sudden it becomes more of an exercise for validation there.
Yeah. And, absolutely, as you mentioned, having a backbone panel saves on time to develop, saves definitely on panel design time, saves in many cases on the data transfer setup time, right? Like you've got all of your reportable parameters predefined. Maybe you add in 3 or 4 if you're adding a marker, and then you're off to the races. So it can definitely speed things up and keep things consistent between different studies within a program. But definitely for any type of quantitative readout, or any type of really sensitive readout, with any flow cytometry panel, anything you do to it will impact everything else in the panel to some degree at the panel sizes that we're talking about. So anything above about, I would say, minimum of 6 colors, then any addition is going to have some sort of impact on the other markers in the panel.
Now, one of the things where a backbone panel may not be useful is if you're looking to do some sort of transfer of the assay. Say you want us to develop an assay for you and then transfer it over to your machine. The reason I say that it may not be the best, it's not going to be horrible, depending on what sort of instrument you have, right? So we've built these backbone panels to work on our instrumentation, our specific laser setup, our filter setup, the strength of lasers. And so we know it's going to work, properly. It's going to work well because we put a lot of time into that. But that doesn't necessarily mean that it's going to transfer over to your machine. So if we're going from a SciTech Aurora that we have on our site and you want to run that on, something else, a Lyric, Fortessa, even if you're looking at a Sony or something, then there's going to be further testing, and you might be better off doing a fit for purpose panel rather than a backbone panel.
Sure. No, and that makes sense. And, in the context of this paper as well, anything else that, would make a backbone panel either more advantageous because you got a starting point, or less advantageous because of some additional type of work that you would need to do based on the paper recommendations?
As far as advantageous, I think it's good because we've already got the expertise for the markers, right? We know that they work. And when you start a panel from scratch, now what we have is our own preferred clones. And so we can more rapidly build something like that. But it is always, if it's new markers, then it takes a bit of time. But as that relates to validation, we've also probably got pretty good ideas in these markers how they're going to perform when we go through the various test scripts for a lot of the matrices.
Okay. And that makes sense. And I guess, Adam, if you were running your own trial program, would you include a backbone panel or would you not?
So the way that I like to think about a backbone panel is that, and this is the same thing with off the shelf, is the consideration, what am I doing? What am I looking for? And so if I'm okay with a two tube setup, I'm probably going to want to take an off the shelf panel and then pair that with maybe a receptor occupancy tube, for example, right? But if it's something where I've got almost all of the markers that I want in this backbone panel, and then I only want to add one or two markers to that are key to my therapeutic, I'll probably just go with that backbone panel, get that finished up, finish up the optimization, and then get it qualified or validated. So I think it really depends on what all you're looking to get out of your trial.
Okay. So to interpret that, basically you're saying not only would you have backbones, you would also have pre-validated panels and also custom validated panels on your trial?
He just sold me on a whole lot of panels. Yes, getting lots of data. But the nice thing is here with some of it, we're obviously moving as an industry towards more and more spectral, right? And so much of this can be handled in a single tube. Now, we've talked about it with RO assays before, that the preference is to keep RO assays simpler, right? Just because of the nature of the assay. And so in that sort of case, then you probably want a couple of panels, right? You don't want to necessarily have a 40 color RO. I'd be really interested to see how many people are doing that out there. And if you are, please drop us a line. We'd love to have some conversations. It's just out of scientific curiosity. I know people are always pushing the envelope and love to see what's out there. But I'm certainly open to doing that multi-tube process there.
Okay.
How about you?
Yeah, I think I would. I think trying to set up a standard within a company makes sense. And as you mentioned, with the advent of spectral, it just becomes a lot more feasible to do quite a bit, right? So that if you have your immunophenotyping backbone, you can add on like whole other subsets potentially with drop-in panels if needed there. So I can see that as being a essential piece of any translational scientist toolkit at this point. Mm-hmm. The only drawback that every once in a while I struggle with is, as we design these, are we losing some type of efficiency? Or, is a panel not quite as good as it is? Like maybe it's like, 90% of the way there, but could it have been 100 if we, made it as a bespoke panel? I think that answer is probably yes, but, how often is the 90% good enough? And I think that is pretty frequent.
Well, and how often does that off-the-shelf panel serve all the needs that you have?
Sure.
So it's a balance for sure.
Yeah. Excellent. Well, yeah, very interesting, both the paper and the backbone panel discussion and the impact of spectral on all of it. So looking forward to getting somebody with a 40 color RO panel. But also in terms of feedback, I wanted to ask our fans for another piece of advice related to these sort of large scale panels. What is the stability for an antibody cocktail in your laboratory, and how do you determine that?
That's a good one. Mm-hmm. Yeah,
Right.
to hear what people have to say on that. And, we are always going out there, conferences, looking at literature. We do our own testing internally, but we're not going to cover everything. And so that's where we'd really love to engage the audience. And, if we like it enough, we have a good enough discussion, then maybe we will invite you on for a show.
Absolutely. Yeah. All right, excellent. And that is, of course, the main portion of our show. So I guess we can move into some Thanksgiving traditions, it being the right season for that sort of thing.
Oh, yes. I have a question for you before you get into yours. So
Okay.
something that has been long-standing discussion with me, and I asked many people of this, and it's amazing how I'll liken it to the whole peanut butter and jelly, right? I think we talked about this before about whether you just use grape or you can use other jellies. And I'm very much a purist. I just want grape jelly and don't give me any strawberry garbage, whatever. Here, is mac and cheese a Thanksgiving side dish?
Now, of course, with my Georgia roots, mac and cheese is pretty much an everything side dish. So yes, we do indeed have mac and cheese at Thanksgiving.
Is it homemade mac and cheese, I'm assuming?
I mean, at the classier Thanksgiving affairs, but with a 5 and a 7-year-old, it is the craft at this point.
Yeah. Now, I will open up that there are other ones. There was a YouTube channel recently that I saw, and essentially going through taste testing of different mac and cheeses. And it actually inspired me to start trying out different ones. So Cabot makes mac and cheese, and they had highly recommended. It was really good. Cheetos has a mac and cheese.
No. Cheetos?
Cheetos. You should check it out. So I haven't actually made it yet myself. I have it in the cabinet ready to go on one of those nights that mac and cheese is needed. But supposedly it tastes like you're eating Cheetos. So for what it's worth, if you like Cheetos, it might be worth a try.
Okay.
For me, I got to be honest, yes, mac and cheese is definitely part of the whole Thanksgiving thing. I mean, it pairs with stuffing and mashed potatoes, and it doesn't seem out of place there. So, but I don't know. I'm not a total traditionalist when it comes to things, so maybe that's where I fit into it more.
Gotcha. Yeah, no, that sounds like a very dangerous hobby, Adam, in terms of trying out different amounts of mac and cheese and different brands of mac and cheese. That sounds like a calorie loaded activity there.
You see, you're mistaking it. So they've eliminated the ones that I know not to try. Now, even they did frozen pizzas, right? And I have always had Red Baron and stuff like that. I'm not a DiGiorno fan. It just feels like it's kind of puffy bread to me. But they mentioned this other one, Table 87. And so I was like, oh man, I went to Wegmans and saw it there and I picked it up. And they're going on in this YouTube video about how it looks like a real pizza. It tastes like a real pizza. I'm like, come on, really? You know, like, were you paid for this? And I will say that I'm not paid by any of this. And yeah, right. I tried it. Dude, it was awesome. Like it tasted legit like pizza. So if you're ever looking for a good frozen pizza, Table 87. But again, I love my Red Barons and stuff like that too. But this was different. It actually you could have confused it with decent pizza.
Huh. All right. Well, now I've got nothing I'm going to go try out.
See, but I have to try all those other ones that they said were horrible.
Okay, the internet to our rescue.
Yes.
And the next question is about how the internet leads to our downfall, I guess. In terms of your favorite or least favorite earworms.
Oh my goodness, I don't know that I can say them. I don't know. There's an adaptation of Frozen where they change the words, and yeah, I won't mention what the words are. They're not all audience friendly, but that one's up there. And I'm trying to think. How about you? What do you got on there?
I mean, what set me off to this question was the title of today's podcast in terms of backbones that connect to more than just the hip and shoulder. Because I did get that song stuck in my head for a solid hour after I came up with that title. So, that's got to be on the list. But in terms of most or least favorite, I'd say the least favorite has to be the Lego song that says that it's going to get stuck inside my head.
Everything is awesome. Sorry, is that the one you're talking about?
No, there's another one that's even worse. I'm not going to say it that way. Like no one on the podcast or our listeners has to have it in there. But yeah, Lego, the Lego franchise is very good at earworms.
Yeah, I think as far as worst ones though, for a long time up there was Baby Shark. So.
Oh goodness.
Yeah, I haven't heard that in a while, so I don't know how much that gets stuck in people's heads anymore.
Well, you've said it, so now it's going to come up on your Pandora station at least once, right? So, yeah, excellent. So, great episode today. Thank you, Adam. And for our next episode in December, we'll have a guest on, Kelly Lunson from Florifinder, and we'll be talking about fantastic fluorophores and where to find them.
Yeah, it was a lot of fun. Thanks for the discussion. Really enjoyed this one. And thanks to everyone for listening. Take care.
Thank you.